Okay, can everybody see my screen? Okay.
Yes.
Hi everyone, I'm Jack. Welcome to your last homework 6, and this is lab 10. A few announcements before we dive into the homework. So homework 6 is due not on Sunday, but on the last day of of this course. So it's Tuesday, April 27, midnight. So you have, I think, 2 more days to work on the homework. And the office hours in the next 2 weeks will be held on regular hours. So Friday, April 9th and April 16th from 4 to 5:30 p.m. and Saturday, April 10th and April 17th from 10 a.m. to 12 p.m. And for this homework, I know people are really from different backgrounds, and some people want more specific help on coding, where some people have difficulty in understanding the main idea and approaches to a question. So I try to take a balanced way. So in this lab, I will mainly focus on the main idea for solving each problem. And I will, of course, include a few codes if that's necessary, and some sample nodes that you'll actually need to use to finish the homework will be provided in a separate file. So a bit of background first. For homework 6, we will be focusing on glioblastoma, and it is a type of aggressive brain cancer, and it starts in astrocytes, um, which is a type of cell that supports your nerve. And where you will be asked to do some simple TCGA exploration. So uh, across 36 cancer types. So for question 1, you are asked to simply just count the number of samples that satisfy a series of criteria. The first criterion is that you need to subset samples corresponding to glioblastoma in TCGA. So it's really easy. Just go to this website and you will see the page here and click on exploration and then cases. So you will find tab for primary site, project, and disease type. And you should click on the correct option that uh, gets you glioblastoma data in TCGA. So for example, like for primary sign, maybe you want to click on a check grade. Uh, and I think you another several other criteria should be like I think gender, vital status, and age. And it's similar idea. You just click on the clinical tab and check male, and I think in vital status you should check alive, and then under age here you just type in the correct age, the range of age the question asks you to subset, and then at this place it will show you the number of samples that satisfy these few criteria. That's really simple. So broad Firehose. So you need to first install the relevant package file browser. It facilitates efficient download of processed TCGA data. So you need to use the Delve tools, but if you haven't installed that yet, you should first use install packages Delve tools and then install from GitHub. This is the vignette for file browser, and feel free to explore it. So question two asks you to get process data of glioblastoma from Firehose. The command you will be really using is just sample.clinical cohort equal to a cancer type. Here I use BRCA as an example, but you will need to find the correct abbreviation. This is abbreviation for a cancer type. You need to find the correct abbreviation for glioblastoma yourself. So the command you will be using should be metadata cohort. You will print out a data frame with two columns. One column is the abbreviation for cancer type. Another column is the detailed description. So you need to read this column yourself and find the correct abbreviation and put it here. It's kind of weird that in this way you just get actually 150 patients, because we are allowed to really just download 150 patients at a time. So to get all the patients, maybe thousands of patients, you actually need to do this in a for loop, like this one shown in the screenshot. So don't worry, I have already provided this chunk of code in the sample code file. You can just run this code, copy this code and run yourself. The only thing you need to change is this abbreviation for glioblastoma. So I won't explain the details of this chunk of code, but it's just a for loop that extract 150 patients each time and store it as an element of a list. And then we use this do call rbind command to row bind every element of that list to generate the final data frame. So for question two, you will get a data frame in the end with patient in rows and a bunch of variable measures in columns. And it asks you to calculate the average age at diagnosis. So you need to find a column that corresponds to the average diagnosed age and just calculate the mean. So the calculation part should be really easy.